Okay, I guess we can go ahead. Hi everyone. Welcome to the third session of the cutting edge techniques and models. And this is Tong from Education Committee. And together with Journal today, we are going to moderate this session, which will be on the topic of genetic tools and mouse models for auditory research. So for those of you who don't know Cutting Edge Techniques and Model Initiative, so this is a new initiative from ARO Education Committee. We'll be hosting discussion panels, which will be very dynamic and interactive, and we will introduce speakers with experience in some uh, doing advanced techniques and or like animal models who are also working in auditory research. And today we're gonna have two wonderful speakers to introduce genetic tools and mouse models for auditory research. The first speaker we have is Dr. Isabelle Roux. She is from NIDCD at NIH, and Dr. Roux is a staff scientist at National Institute of Deafness and Other Communication Disorders at NIH. She is an expert in human and mouse genetics, cochlea physiology, and auditory biology with more than 2 decades of experience using advanced genetic imaging and physiological tools to study hearing under both physiological and pathological conditions. Her recent research focused on the role of endolymphatic sac in inner ear development, the genetic architecture of hearing loss, and the generation of function characterization of genetically engineered mouse models to uncover mechanisms leading to hear loss and support preclinical therapeutic development. She has led collaborative genomic studies in understudied populations, including the Yoruba community in Nigeria, and is currently co-leading a similar initiative to define the cause of childhood hearing loss in Puerto Rico. She is board certified clinical laboratory genetics and genomics. We also have Dr. Lijin Dong, who is from National Eye Institute at NIH. Dr. Dong is an associate scientist at NEI and a director of NEI Genetic Engineering Core. He specializes in rodent genome engineering, cellular reprogramming, and stem cell differentiation. Dr. Dong and his team are responsible for generating genetically engineered mouse and rat models that support research across NEI and multiple other NIH institutes. His current research focused on DNA repair responses in mouse zygote following CRISPR-mediated genome editing, with the objective of improving precision and efficiency in engineered animal models. So, without further ado, I will let Isabelle take over for the first part of presentation, and then followed by Dr. Dong's presentation, and then we will have a interactive discussion after their presentation. If you have any questions or any technical issues when during this session, please go ahead and reach out to headquarter@arrow.org. And we encourage people to ask questions during presentation. So please leave your questions in the chat, or you can also raise your hand and just unmute yourself after the presentation to ask questions for our speakers. Okay, I will let Isabelle take over.
Good afternoon. Thank you so much, everyone, for attending. And it is really a great pleasure and honor for me to lead this session with Dr. Lijin Dong, who is Lijin is a long-term collaborator, and I'm very excited to hear all the new advances in CRISPR technology is going to share with us today. But first, I want also to thank Dong, Jarnell, and Kathy, and the committee of ARO for extending this exciting opportunity for telling you more about the highlight of the advance in mouse genetics, which are really shaping our study of hearing. So I'm going to share, if it's okay with you, and then dive in. We are going to, may I ask you if you see my screen?
Yeah. Yes.
Oh,
yes.
Thank you so much. So today we are going to highlight some of these advances in mouse genetics. And we will have mainly two parts, one focusing on classical approach of mouse genetics, which have been constantly been improved by investigators and fine-tuned to allow you a better precision of the manipulation in mouse genetics to study what you're interested in. And then Lijin will take it away and tell you about all this exciting new technology of CRISPR-Cas9. So why are mouse models central to hearing research? Well, I think one of the key parts is that they have a shared cochlear, there is a shared cochlear structure and genetic homology between mice and humans. In fact, it was really interesting way back when the research in human genetics of hearing loss started, we actually really learned from spontaneous mouse mutant and then mouse which were being engineered. And in this kind of an overview from Carson and Abraham, you can realize that at least 27 of the genes which are now known to be involved in human form of hearing loss, were actually first identified in mice as important for hearing. And then almost all the genes we identify in human genetics, we try to do mouse models to better understand the pathology. But remember, at the beginning, we had these just lot scores, and it was really complex. And we had just big region of DNA where the real gene who was causing the phenotype was unknown. And really having information coming from the mouse was really critical to help us identify really the accurate source of the hearing loss. So mouse have been shown to be suitable for studying congenital, progressive, noise-induced, and age-related hearing loss. So they are really great models. In every technique, I always kind of try to also know the limitation. And although I'm totally excited about mouse genetics, I also want you to realize that really the auditory threshold curve for mouse, which is here, is really shifted as compared to human. Human auditory threshold curve is here in bold. And you can see that gerbil, for example, or rat are much closer to the human sensitivity of hearing. And we're going to talk about it a little later, maybe as a conclusion, because there are now new tools which may allow you to also modify the genome of the gerbils or other system. So why has been mice study so central? Well, first, you have to consider their small size, of course, and their relatively shorter lifespan than humans, so that you can do, you can study different generation of mice. That's one thing. But the big thing is that there is a variety of mutant mice available and new genetic tools and approach which allow genetic modification that can be cell type and time specific. So it's really a lot of possibility in terms of mouse genetics to study hearing. There is, whoops, I apologize. Do you still see my screen or?
Yes.
Yeah.
Sorry. So I have lost my screen, but there are also some preclinical, so these mice are also really helpful in in being used as preclinical model of human form of hearing loss to develop or validate therapies. Again, limitation, there are some limitations because sometimes these mice don't quite reproduce the phenotype seen in patients. And these different may come from the different changes in hearing that may be different and affect different frequency in human and mice, or the intensity of the hearing loss may be different in human and mice, or it may be some syndromic manifestation. You know, sometimes you have hearing loss associated with other phenotype in patients, which may or may not be seen in mice. For example, I work on pendrin, and the mice don't have a goiter, whereas it's present and associated with pendrin mutation in human. So some limitation, and we're going to go back to this. So despite this limitation, I really think that mouse genetics has really helped almost every side of the research in hearing during these last decades. I really see mice as building blocks, which are really helpful and cornerstones to our research. And here it looks like a long list. It was just to formalize and describe to you all the potential application. And I'm sure every day there are some very smart scientists who develop some new application. But just whether you are an embryologist, a human geneticist, an electrophysiologist, mice can really help your research. So these mice can help you assess importance of a gene or pathway in hearing, trace lineage of your favorite cells, eliminate them with some very cool system for example, expressing diphtheria toxin or sensitivity to diphtheria toxin. You can study the pathogenicity associated with a gene change and test treatment. You can use reporter approach to study the expression of a gene, silence its expression either constitutively or conditionally at a given time in all or a subset of cells. You can generate preclinical model for developing and validating therapy. Now, if this is really not your cup of tea, you can also identify and visualize specific cell type to study in wild type and pathological conditions. For example, for a long time, I was trying to find some specific marker of developing efferent in the very young cochlea. And all the antibodies I was trying were both labeling efferent and afferent, which was making things really difficult. And then I was able to, after screening different methods for reporter, I could identify a chat cray, which was allowing me to very specifically and finely label the efferent fibers and see if in my mutants there was a change of efferent innervation or not. So this is the same approach has been used a lot by also electrophysiologists or some of you which are really finely studying the structure, for example, of the nerves, or trying to, in these old bundles of nerves, which are so difficult to study one by one, you can do sparse labeling, for example, which really allow you to see and follow one fiber, which is really helpful. You can also study protein localization and regulations, for example, by inserting tags. For a long time, we were trying to, with a very talented postdoc I was working with, Dr. Yunjie Lee, we were trying to visualize the change of expression of pendrin upon different conditions. And it was really hard because we had a really good antibody to detect the protein, but we couldn't know if it was inside the cells or outside the cells. So we developed some mice with a tag in the extracellular region of pendrin with Lijin to really be able to follow the change of expression of the protein depending on the treatment. You can also activate cells nowadays and assess their activity using optogenetic and chemogenetic tools, and also detect transmitter release, for example, with different sniffers for neurotransmitters or pH sensors. So really a wide variety of tools that you can use. So now I don't want to, unfortunately, we don't have time to really go through the whole story of mouse genetics, but I just wanted to emphasize that by these beautiful images from Charles Babinet from more than 30 years ago that the initial modification in mice have been done by transgenesis, by DNA injection into the pronucleus of the zygote. And at that time, selection was impossible, and there was a random integration of your transgene. So you may still have this approach when you are talking about back transgenic. In general, this is this approach which is non-targeted. But most of what I'm going to tell you today about is about targeted transgenesis using these embryonic stem cells, which have been so helpful to the expansion of mouse genetics. Because using these cells, selection is possible, and targeting is possible using homologous recombination. And then Lijin will tell you more about CRISPR-Cas9 editing, which revolutionized a lot of these approaches. So what are the main targeted genetic modification you can do with classical genetics in mice? Well, there was insertion of a reporter. So here the idea is to use the promoter sequence and the enhancer and all the machinery of expression of your gene, but instead of having your gene expressed, monitor its expression using some form of reporter. So you can follow where your favorite gene is expressed. Another way to study your favorite gene could be to add a tag at the end of a protein, or it can be fluorescent, it can be a small HA tag. So that was one modification we could do. We could also modify gene by doing some genomic deletion to obtain a knockout or create a conditional knockout. And we used embryonic stem cells to select very rare events of homologous recombination, allowing this insertion of a desired modification in the mouse genome. And after that, we used to reimplant these selected ES cells into blastocytes and really hope that they would colonize the germline, so that from these mosaic mice, this is the first mutant mice of autophaline, then I worked on, you were really hoping that the ES cells would colonize the germline so that the modification could be passed to the next generation, and you establish a line. So one of the limitations of this was that at the time you could only do it on specific mouse strains, and certainly not in other species. Progressively, it was feasible in rats, but essentially people were really focused on mice. So doing this takes a lot of time. As you can see, it can take 8 to 10 months, and cost is quite costly. So if I was you, if you are starting a new project and you have some idea of the mice you want to use, I would definitely first check if the mouse I need is already available. And these slides will be shared, and I put you here a number of resources that I very often check because they are really helpful for identifying some precious lines, or also identifying investigator which may have these lines. Some of them are available through depositories, facilitating their exchange. So the key tools to manipulate the DNA has been these site-specific recombinases. So I'm sure you all know about CRE-LOX system. So CRE is a site-specific DNA tyrosine recombinase derived from P1 bacteriophage. It catalyzes DNA recombination between two 34 base pair recognition sequence referred to as LOXP site. And each LOXP is composed of two 13 base pair palindromic sequence that you can see here. Palindromic means that if you read 5' to 3' here and also 5' to 3' here, you will read the same thing, which is pretty cool. But the important thing, besides the integrity of this LOXP and its sequence, is its spacer region or core region here, which is really important for the orientation of the LOXP site. So upon binding to the LOXP site, the CRE-mediated recombination will result in cleavage of the DNA and reciprocal stand exchange between the two LOXP sites. But the key is the orientation of the LOXP site. So if the LOXP sites are located in the same direction, meaning that their spacer are in the same direction, then upon action of the CRE, you will have deletion of the fragment which is between the two LOXP sites. However, if the location of a spacer region is inverted, you will have inverted LOXP sites, and there will be a constant flippage of the region between the two LOXP sites upon action of the CRE. Okay, and I'm going to tell you in a minute about a trick to make this stop and take advantage of that. But if you are just trying to delete the region between the LOXP site, the last thing you want is this. So just be careful about this when you receive mice which haven't been tested yet, but the LOXP sites are in the good orientation. And then in terms of another modification which can be done using this LOXP site is this very large translocation from one chromosome to another, for example. There is not just one recombinase, there is several types. Another one very popular is FLP for flipase. It's a tyrosine recombinase also, which mediate the DNA recombination between 234 base pair recognition site, referred to as FRT site, just like the LOXP. And another system is called the DRE recombinase, which works with the ROX site. Why is it interesting to have different recombinase? Is that they are in general really specific to their recognition sequence. So you can mix and match and really do some gorgeous modification. What is key to know is that the CRE will have different activity depending on the sequence of the LOXP site. And in general, if you have different type of LOXP site, the recombination will happen between two LOXP sites which are identical. And there are a few exceptions that we could discuss another time. But what you have to keep in mind is that if you want to have recombination, it will be between two LOXP sites, which are identical. But there are some variation, as you can see here. And then same thing for the flipase, there are at least 3 sites which are recognized, and then the DRE. Okay, so what are the applications of this? If you mix and match, you can actually do a lot of cool manipulation genetically that you couldn't otherwise. And you can do all of this in vivo or in vitro, but you can also do them in vivo. And that's a really beautiful system that Dr. Joshua Sainz and his team took advantage of, this multiple LOXP site and their specificity to generate, for example, these gorgeous rainbow mice, which allow you almost to label each individual neurons expressing a different color by combining the fluorophore that are expressed, the reporter that are expressed in each neurons. So I wanted also to give you some other application of this system of 2 locks, and this is one of them, the flex or flip excision switch, also known as double flux inverse orientation. So I'm very interested in this because, you know, in many ways, we are often trying to knock out the expression of a gene. But as we are moving more toward gene therapy and ways to restore a functional phenotype, it can be really interesting to try to see if you re-express the gene at different times, for example, or in different cells after its initial absence during early development, could it even allow the phenotype to be rescued? And that's one system where you can have silence expression of a gene, let's say the gene R here, which initially is not expressed because with this construct, you only express GFP first. But notice that you have different LOXP sites, which are in inverted orientations, okay? And there are 2 sites. Look at the one here and here, which are the same, and this LOX 511 here, which is different. So I told you, when you add Cray in this configuration, there's going to be a flippage. And the one which worked the most efficiently is going to be just a regular LOXP site. So upon action of the Cray, you will have inversion. You can have inversion of the gene this way. But then, look, you change the orientation of the LOX511. So because now the two LOX511 are in the same direction, if Cray acts again, it can delete the GFP reporter. So from a cell which was expressing GFP, you now are able to rescue the expression of your favorite gene. Okay, so I think this can be really helpful to make a conditional rescue of a gene knockout, especially if you think about it and the different tools which are now available as viruses and how you combine both. You can also introduce a conditional point mutation or functional mutation in vivo by this approach. For example, by having here a wild type form of your favorite exon and here a mutated form of your exon. So upon action of a Cray, you can have then expression of the mutated one or vice versa. So, and it, for electrophysiologists, it can also help the spatiotemporal expression of optogenetic reporters by ensuring that the reporter remains silent until a cell-specific Cray is provided for optogenetic reporter delivered by an AAV approach. So quite a nice way to induce the re-expression or allow the expression of a gene. There are some drawbacks, though. The system can be leaky or have mosaic expression, so you have to check. But this is, I think, a very nice application of this multiple LOXP site technology. Another one I wanted to mention, because you may find these mice helpful, is this systematic genetic modification pipeline from COMP or UCOM, and the mice are now developing by the EMA consortium. And the idea was to do a systematic knockout or systematic modification of each gene in the genome, so that can help researcher have access to these different mutants. And really the very nice idea here is that using one allele, which the author called the knockout first conditional allele, you could, by successive action of flippase and Cray, have both a reporter allele, a conditional knockout, and a total knockout. So how does that work? Well, as you can see in this construct, the exon 2 of a gene was flanked by LOXP site, and there was also a neomycin cassette, this is really a usual trick to select the events of integration in the genome of your construct. Then the author also inserted a LAGZ, which codes for beta-galactosidase, and this is a very traditional reporter system, because LAGZ will express the protein associated with the gene LAGZ is beta-galactosidase, and if you give beta-galactosidase a substrate, which is called XGAL, you can have production of a blue precipitate where you have expression of your favorite gene. So that's really the idea, because here, if you have FRT side boarding this construct, and you breed the mice which have this allele with mice which express flippase, constitutively and is particular in their germline conditional out due to the recombination between the FRT site and elimination of the fragments in between. And that's what you can see here. And then if the Cray recombinase act, then you have a total knockout. Same here, if you breed the mice expressing this construct with a Cray, a germline Cray, then you can have excision, usually between the further LOXP site and creation of a reporter allele. So with one allele, you had 3 different alleles. And I just indicated here for you some reference for the germline Cray and flippase in case you are interested. So that's a very nice system. And if you ever see your favorite gene with a TM1A name, or TM1C, 1B, or 1D, this refers to this, or a variant very close to this. So now this CRE system really allow you some cell type specific manipulation in vivo. And it can, at least initially, I think a lot of us were very happy because it could help you avoid embryonic lethality if your favorite gene was necessary for survival in the embryo. You can also identify cells expressing your favorite gene. You can identify cell type specific role using a conditional knockout allele of your favorite gene, using this specific CRE, and then label a specific cell type by expressing a reporter protein. So just in case it's helpful to you, I have put here some points to consider when selecting your cell type specific CRE. You want a tight expression of the CRE so that you don't have expression unless you want it, basically, unless it's under the promoter that you have chosen. You want a high efficiency of recombination in specific cell type that you have chosen. You need to think of when my CRE is going to start to be expressed, because it's not because it's expressed in hair cells that is going to be necessarily helpful to you, for example. But you need to see, well, when does it start to be expressed? Because my effect mediated by the CRE will not happen before the CRE is expressed. So another consideration is, should the male or dam parent carry the CRE? There's a beautiful work from Liu and colleagues who are talking about this in more details. And then there are possible difference in efficiency depending on the target. I know that I love fluorescent reporters, but they are often inserted in a locus in the genome, which is called ROSA26, which is very accessible for CRE recombination, but sometimes your favorite gene doesn't have this accessibility for the CRE. So you always need to verify that what you want really happened, and that if you are trying to delete a fragment of a gene, to lose the expression of your favorite protein, you need to verify that it's indeed the case, because sometimes the CRE can be really efficient on the reporter, but not so much for your personal favorite target. Okay, you need to also try to see if there is a phenotype associated with the presence of the CRE itself, not to have confounding factor, and also remember possible expression in other tissue than the ear. For a long time, I work on alpha-1 nicotinic acetylcholine receptor, and I try to use CRE which are very specific in the ear and gorgeous, but are also expressed in the diaphragm. So basically, my mice were dying at birth because they couldn't breathe, because they needed the alpha-1 nicotinic acetylcholine receptor in the diaphragm to survive. So just good to remember. Another advance really to this CRE system has been the cell type, but also time-specific manipulation in vivo they have allowed. And one way to do that is to use in vivo stereotactic injection and viral expression of the CRE. And in this case, the viral tropism will contribute to the specificity of the CRE expression and subsequent modification. There are also some very nice inducible recombinase, in particular the CRE-ER and its optimized version called ERT2. In this system, the CRE is fused to a mutated ligand binding domain of a human estrogen receptor, resulting in a tamoxifen-dependent CRE recombinase that is activated by administration of tamoxifen or hydroxytamoxifen. So very helpful if you are trying to control the onset of the action of the CRE. Another one we will not talk about for interest of time, but which is really nice too, is the tetracycline-dependent expression that can be activated upon administration of doxycycline. And then there are some better and better split recombinases, which are all concurring to increase the specificity and the time dependence, because what you really want is to finally control the timing of expression of the CRE and its expression. So I just put here a couple of figures to remind you how CRE-inducible, how tamoxifen CRE-inducible CRE work. Sorry. So what happened is that in absence of tamoxifen, your CRE may be expressed depending on the cell type and the onset of expression linked to this promoter, which are governing the expression of the CRE. But in this case, the CRE is not in the nucleus, but stay in the cytoplasm. So there will not be any recombination. However, upon action of tamoxifen that you can inject or have a pop eat, basically, then there can be a binding to the CRE-ER, and you will have an export to the cytoplasm. And then the CRE becomes active and will work. And in this system that we call lock-stop-locks at the rosa 26 locus, what you will have is the excision of a stop, and you will be able to have expression of GFP. And just one thing I want to remind you is that once this CRE has acted, then the change is in most cases irreversible. So all the cells, whether or not you inject tamoxifen anymore, this action will have been done. And so all the cell will have lost this lock-stop-locks to only express GFP. Okay, so just to answer our moderator's suggestion, I indicated here a list of very helpful CRE to study the inner ear. And as time goes, they are better and better characterized. Here I put the PMID for the author who first published these mice and created these mice. But over time, there is better and better characterization of the expression. And what is also really nice is that as we progress is our knowledge of the transcriptome for some of these cells, they are more and more precise CRE, which can be used. For example, here you have the CALB2 and LYPD1, which can be used to see different type of sphere ganglion neurons, which is really cool. Okay, just one word on RiboTag. This is a really nice system which has been used for tagging the ribosome to analyze the cell type specific mRNA expression in vitro. This was done thanks to this mouse mutant, which at the RPL22 sites have their exon 4 last exons surrounded by LOXP site. And then just afterward, there is the same exon, but with an HA tag. So when you breed these mice with mice which expressed CRE in your favorite tissue, you can have recombination here, excision of this exon 4, which is not tagged, and then expression of the tag exon 4, which will lead for the polyribosome in the cell expressing CRE to have an HA tag that you can then immunoprecipitate to then study. So this allows you basically to concentrate into the mRNA from cells which expressed CRE. And so you can identify, even if it's a small population out of a big tissue, you increase your sensitivity for specifically detecting transcript for this very specific cell type. Okay, so just coming back quickly to this idea of the difference of the comparison between mouse and human phenotype. I told you that it's not quite the same phenotype sometimes in mouse and human. And just here, a very nice example of this, where when the author directly inserted one of the most frequent mutation in pendrin in the mouse genome, one of the most frequent mutation in humans of pendrin in the mouse genome, there was no phenotype whatsoever. But they developed these very nice mice where you have silencing and no expression of the endogenous pendrin. However, they made the mice express the human form of pendrin, and in this condition, they could identify a phenotype. So just lots of different approach, including humanized mice, to be able to study human mutations. So with this, I would like to leave the floor to Dr. Lijin Dong. I would be happy to answer any question at the end.
Thank you, Isabelle. We have just like less than 20 minutes left, so we get to go a little bit faster for the second talk.
Okay, so go first. Can you see my Isabelle? Could you de-share your screen?
Please stop sharing, Isabelle.
Let's see. Yeah. Okay, let me share my screen first. Okay, here we go. Now, can you see my screen?
Yes.
Sure. Okay. All right.
Please go ahead.
Okay. Okay. All right. All right. Isabelle, that was a really wonderful talk. And then she, in her talk, mentioned basically three categories of mouse models, which are important, not only for auditory research, but also for biological research overall, right? Such as in ocular research, which is the field I am in, essentially. So I'm going to focus on my talk on the third category, that is genetic engineered mice, and the most advanced way of making those genetically altered mouse tools, specifically by focusing on CRISPR-mediated genome editing technology. I'm going to perhaps be really brief because we're running out of time. But first of all, I still need to share this piece of history, right? In the work of genome editing that came from Maureen Jason's lab in the mid-90s, where she and her colleagues showed that if you want to perform any kind of genetic engineering, genetic modification to a specific site, there are two things that needs to be considered. Okay? One is really a presence of a double-stranded DNA break at the target site. The other is the subsequent cellular DNA repair response. So two of them together going to determine the outcome of your editing effort, essentially. Okay, so that's that. Oops. Problems. That's not, oh, here we go. Okay, so I'm going to show a few examples here, which are from different cellular different cell types to give you an appreciation what I meant by the importance of the presence of double-strand break and also the interactions of that double-strand break with cellular DNA repair machineries, right? So the first example here is a single DNA double-strand break induced at the target site in mouse zygotes by CRISPR technology, and then allowed F0 mice were allowed to be born, and genotyping performed using tail biopsies crossing the target site, the cutting site. Okay, so this basically is a litter of F0 founders essentially. So you see all those, you see those bands, PCR bands. Okay, wild type bands here. This is the size of wild type and wild type. And number 37, you see 3 different bands here. So there's a wild type allele. There's obviously insertional allele and a deletional allele. So similarly to others, similarly in other founders. But in 77, you only have 2 alleles, right? One wild type allele, one insertional allele. So what's going on there? This is because of the cellular response towards to the DNA double-strand break called indel mutagenesis, right? Essentially, is a quick way to religate DNA, which we'll go through this in a little bit detail, and then but not perfectly with small insertions or deletions. So that makes it useful because if you have insertion or small deletion, you can throw out open reading frame and generating a knockout allele, essentially, right? So, but I want to point out here that some has 2 alleles, wild type plus a endogenesis alleles, and then the others has both insertion mutant, basically 3 alleles. So that is an indication of a mosaicness. You probably heard mosaicness. This comes from the fact that the genome editing takes place in perhaps in this case number 37, not only in the 1 cell stage of the embryo but also in the 2 cell stage, 2 cell stage embryo separately, where you have a insertional mutation and deletional mutation separately, right? So it's a mixture, and in F0 generation, the goal is to really separate those mosaicness by crossing them to the wild type partner, and then in the germ cell pools, so they become pure. So you can separate those alleles, right? So that's why it's important to do a germline transmission crossing, right? So that's that. And then second example is if you have 2 different guide RNAs, then you're often generating a genomic deletion. So this is usually a very efficient operation as well. And then if you change the cellular type, in this particular case, we use the guide RNA Cas9 plus Cas9 protein carried AAV virus and then injected that into subretinal space, and then the primary result of that single DNA double strand is again endogenesis, as shown here. There's 1 base pair or 2 base pair insertion or deletions. So we have seen so far in mouse zygotes in retina. And then if you hit them with a single strand, with either 1 or 2 double strand break, generating a 1 or 2 double strand breaks, in the genome, and then the primary response to that is endogenesis. Okay, and then this is mediated through NHEJ or MMEJ, which we'll talk about that a little bit later here. In fact, here. So what happens that if you have a double-strand break, and then through NHEJ pathway, the free DNA ends is quickly attracting Cu70, Cu80s, and subsequently create a cascade reaction of DNA religation through the DNPK pathway that's called NHEJ. And then alternatively, a different pathway can kick in that's called microhomology mediated regeneration, which can most of the times will generate endogenesis, just as in NHEJ, but sometimes it can be troublesome. It can lead to large deletions or chromosomal translocations. Okay, and as shown in this case, now this is a mouse ES cells, embryonic stem cells. Again, each of the panels here shows a single double-strand break cut. And then the results of that cut is quite different from what you've seen previously in mouse zygotes or in the retina. Good portion of the cells undergo large deletion. Okay, it's not like a quick religation. It's rather it's a quick ligation. It is a large deletion, and then this tells you that the MMEJ DNA repair pathway kicks in this particular cell type. Okay, and then if your purpose is to do insertion, again, you have double-strand break. You want to insert something specifically at the breakage site. For example, a reporter, then you need to utilize the HR pathway, homolog recombination pathway. Again, this is in mouse zygotes. As you can see here, the efficiency is quite high. Okay, the homolog recombination pathway here is quite active in mouse zygotes here. Again, validated by the phenotype here. You have this reporter light up in the target cell type. Again, homolog recombination along with another cell pathway called single strand annealing pathway is responsible for insertional events here. Okay, we want HR. We do not want, as I say, similarly to the MMEJ, is also sometimes leads to large deletions and chromosome translocations as well. The take-home message here is that we need permissive DNA repair environment. A permissive DNA repair environment in our target cell type is important to achieve the goal of genetic engineering. Of course, the ability to generate a double-strand break at a given target site is important here, right? But how this is achieved, we haven't talked about how we reprogram the substrate specificity to generate a double-strand break in the genome, right? So particularly in CRISPR-Cas system, this is achieved through RNA-guided and endonuclease activity, essentially. And this is nothing really new. Nature has repeatedly invented RNA-guided defense systems. Here, I want to make an analogy between Cas9 system and siRNA RNAi system or siRNA system in eukaryotes. So both requires a RNA, a piece of RNA, serving as a guide, searching for the target in the genome, right? So, and then in eukaryotes, in eukaryotes, siRNA guide the risk complex to silence RNA, while in the bacteria, guide RNAs guides Cas9 proteins cut DNA. So both systems use the RNA guides to scan through the potential genome for matching sequences. Okay, now important for in the CRISPR system that appropriately design your guide RNAs, right? So showing in here, for example, in SP Cas9 here, use a PAM sequence of NGG, right? So that probably will work for GC-rich contents. While if you have a AT-rich contents, you couldn't find a NGG PAM, then in that case, you probably want to go to the Cas12 system, which use a different PAM sequence, TTTN, for example, here. And so with the availability of a variety of different camps, you can almost design a guide RNA in a given side within the mammalian genome here. So I've listed commonly used online tools for guide RNA design here. Okay, now the typical workflow here. Essentially, you produce the guide RNA by in vitro transcription, and then you incubate the guide RNA with Cas9 protein in this case, and form a RNP ribonuclear particle that is a Cas9 RNA loaded with guide RNA, and you through one of the three ways you deliver that into mouse genome. Showing here, the first here is microinjection, or you can do electroporation in vitro. That allows you to process the delivery, let's see, 50 or 100 to 100 fertilized eggs at the same time. Right? A lot easier than microinjection. Alternatively, this is IGONA method. That is in vivo electroporation. Essentially, you don't have to isolate the eggs anymore. You open up the ovary of a female and then electroporate directly through the oviduct and then deliver the CRISPR reagent directly there. So the advantage of that, this is a highly simplified way of performing genome editing because we don't have to isolate the eggs. That's make it useful for species that we do not know how to manipulate their eggs. For example, we do not know how to isolate their eggs, and we don't understand their reproductive biology very well. Showing examples, ground squirrel, tree shrews, hamsters have all been genetically modified through IGONA methods. Okay, and then so I'm going to skip this slide, just showing genome editing with CRISPRs, what we can do essentially, and then for the next generation CRISPR technologies, which includes prime editing, base editing, and CRISPR-I, CRISPR-A. I'm going to go through this very quickly, just by showing you a project that we have done. Okay, this is a base editing, and the goal is to convert this C residue to T, generating a TAA. TCA become TAA. That's a stop codon, right? And then generating a premature stop codon here. This is one way of generating a null mutation or a shortened version of a protein, essentially. And as shown in the bottom panel here, we have our target residue converted, but I want you to pay attention to the last residue within this window here, that this is also converted. That is because within this auditory window here, there's a C and there's a G. On the bottom strand of G is a C, right? So it gets to be converted regardless by this base editing, but this base editor, does not have the ability to differentiate. It does not know which ones is the one that you intend to change. Okay, that gave the problem of bystander editing. It just wants to point out the limitations here. However, in this particular case, we want to produce a TAA premature codon, and then the bystander editing here is behind this stop codon. So it did not really matter. But here is the idea. So when the the knee case binds to the target, and then generating a bubble, right? And so the melting locally, the double strand melting locally exposed a few base pairs to the machinery here, to the deaminase here, and the rest of residues are not accessible. Okay, so the few base pairs that's directly exposed to the auditory machinery is called auditory window. So within that window, if you have another residue, another base pair that is exactly the same as the target base pair as intended, you get both edited. Okay, that's the limitation. Okay, all right. So we often worried about the off-target effects in CRISPR mediated genome editing. But from our experiment, from our genome-wide sequencing analysis and others as well. So particularly in mouse zygotes, that's not really an issue as showing here. So this is the control. This is the experimental group. So as long as you titrate your guide RNA to the smallest dosage that's effective, and then as long as you design your guide RNA appropriately, that's not going to be a huge issue here. So that's the message I want to deliver here. And also, the last thing I want to tell here, again, a revisited topic of permissible cellular environment for genome editing by throwing a dramatic example here. So here is, let's see, pre-oocytes. As you know, this is in embryonic stage, where pre-oocytes get rested right after meiosis 1, right? And then during meiosis 1, important physiological events that takes place is called crossover. I'm pretty sure you all heard of that. During crossover, maternal and paternal chromosomes exchange genetic materials through hallmarks recombination. So this is actually induced by double-strand breaks, as shown here. So it's a staining, immune staining of double-strand breaks, and then it's mediated by this enzyme called SPO11. It's only expressed in pre-oocytes. And then other DNA repair mechanisms, such as NHEJ and MMEJ we mentioned, are downregulated or excluded even from the nucleus during meiosis I such. So in meiosis, cell maintained a HR or hallmarks recombination specialized state. It's only permissible, it is a highly permissible cellular environment for hallmarks recombination, okay? And then in zygotes, later on in zygotes, however, things changed. So the DNA repair environment switched to include NHEJ and MMEJ, but overall remains poorly defined. So this is really what we're interested in. We have several research projects focused on this point, basically. And I basically want to end my presentation here is that CRISPR-mediated technology enables a programmable introduction of double-strand breaks at the site of recombination, desired site of recombination. And then it is really the interaction of the cellular DNA repair machineries to the double-strand breaks that determines the outcomes. And then in the next generation of maybe gene editing approaches, we probably should pay attention to the cellular, how permissive it is of the cellular DNA repair environment, and how to regulate that environment towards the goal of mutagenesis. So I'm going to end here, and I'm ready for questions. Oh no, I'm going to hand this back to Isabelle, so she will wrap up the presentation. Thank you.
Thank you, Dr. Lijin Dong, and thank you, Dr. Isabelle Roux, for this wonderful talk and for sharing these insights. I would encourage audience to ask questions because I don't see any questions in the chat box yet. So please feel free to unmute yourself in case you have any question. We can still take 1 or 2 questions. All right.
Please also feel free to email us if you have any questions afterwards. We will share our slides and you will have our emails on the introductions.
Yeah, that would be great. So I have a question regarding CRISPR-Cas9 technique about like how cost effective it is or compared to traditional methods of gene editing.
Say that again, I'm sorry.
Cost effective?
Cost effective. Yeah. It's probably much more cost effective compared to traditional gene targeting. For example, here, this is done, number one, this is done directly in mouse zygotes versus that embryonic stem cell stage is necessary in the traditional approach, right? That alone save time, save money. Yeah, it's much more cost effective. Okay.
I have a question for Lijin. Do you think we are going to change our favorite animal models, maybe? Because now with CRISPR, we can do modification in any strain, basically. So
yeah, Yeah, that's a great point. Because of the simplicity of the technology, especially with iGonads, we don't have to learn the reproductive biology from each strain of interest, right? Essentially, we just open up ovary, the female, open up the female exposed ovary, and then just electroporate genome editing reagents into the ovary, and then let it generate the mutations of desired mutations there. So, yeah, yep.
It's very exciting. Thank you,
Thank
Lijin.
you. Mm-hmm.
There is a question, if gerbils are more closer to the humans, why don't we use them for the gene editing or other genetic manipulations?
It's gerbil. Could you repeat that again? So sorry. Can you read that question again?
Sorry.
If gerbils are more closer to the human genome, why don't we use them for gene editing? Basically, like, can we use gerbils in place of mice because they are more closer in terms of auditory response?
The simple answer is yes. Gerbil has been used to perform CRISPR-based genome editing. Yes, I think you can expect to see more and more gerbil models become available.
Great. Is there any other question? Anyone wants to ask question before we conclude the session?
I don't see any questions in the chat. So I guess we already ran over 14 minutes. So I guess we can conclude the session. And thank you, Dr. Dong and Dr. Roux, again very much for a wonderful session. And thank you everyone for joining us. We will share the resources and the slides with the recordings later in our YouTube channel and the website. So yeah, I guess that's it. Unless anyone want to say anything else.
Thank you
Okay,
so much, Dong and gentlemen, for having us.
We have a slide about the tips for like how mouse genetic background can affect your study and what are the important points to consider, which Isabelle could not share with our audience here, but in the final recording there will be that slide probably, so then that will be helpful to go and check.
Yeah.
With pleasure, and also an invitation for anyone interested who would like to know more about auditory research. Every year there is a class organized at NIDCD, and it's called Ear Essential, and you will also get some information about this. It's a totally free course lasting about a week, but it's very exciting about the latest of hearing research in different approaches. You're all welcome to join.
Okay, same here. And if any of you have any questions regarding the CRISPR-mediated DNA, CRISPR-mediated genome editing, feel free to send me an email. I see if I can answer them. If you need help, technical, maybe information, so feel free to send me an email. Okay.
Wonderful. Thank you both. And I think we will end here and hope everyone have a wonderful day.
Thank you.
Thank you.
Bye-bye.
Thank you for joining.